I have taught you some, and I just motivated you. What is our ambitions? What is our goal for this course? And I try to give you some idea. What is the biosensors? How we can make a miniaturized sensors, a small device, so that you can bring this technology in the market. And I just told you about the some future perspectives for this area. So this is not only a kind of course. This is also kind of your future plan that you can make from this. So, and this is the main things that breakthrough discoveries: don't keep your innovation inside the lab, bring it outside for mankind, right? This is very important line. Okay, now let's start some new topic that, hello? Yes, is it audible? Are you asking anything to me? Okay, everything fine? All?
Yes, sir.
I think someone
Yes,
asked, I think someone asked me anyway. So let's start the new topic, that is the translational health research. So what is this? And why I want to uh, show you something about this topic, because it is very much related on this current condition. Also, you should know why we should learn this for the sensing device, also for bench to bedside diagnosis. So I'm just always saying this bench to bedside, right? Uh, So if you see that, I always mention this one, that bench to bedside. It means bench means the lab that you are developing your idea, and bedside means near the patient or the end user they can use for themselves. So if you see here the definition, that translational research, it means the laboratory, whatever you are doing. And so generally we are testing, we are developing using some high-end instruments, some benchtop, very big instruments, sophisticated instrument. But that after that, maybe you just made something. Maybe you are doing some animal model, you are validating your data, right? And then finally you are developing something. What is the something? Maybe some drug, some medicine, some device, and whatever. So you are developing inside the lab. Then you have to go for the human trial finally for approval to bring to the market. So you have to go for the human trial, right? That's why we waited for a long time for our COVID vaccine. Although I can say it is not so long, it is too so short time, I can say, because for development kind of drug or vaccine, it takes long time. So this is the translational, means you are translating your idea for the patient, near the patient they can use. So you can see here that it is kind of the not the unidirections, it can be the both directions. Why I'm saying here, because it is included like some basic science, some clinical research, right? Basic science means, in the lab whatever you're doing, then you have to go for the trial. First trial maybe the animal trial, then go for the human trial. But when you come to the human trial, that time you may face some challenges. You may face some side effects. You may face some problem that may not work, or they're working with lots of side effects there. So you can't apply then this innovation, these things. Uh, that is fully true for biosensor device case also. So what then we are doing then? If you face any challenges, what do you do? Then go back to the lab again. That's why I'm saying it is not the unidirectional. You have to come back again to the lab and solve this problem. Again, you have to check for the human trial. So if that's why it is not so simple and not so easy, not so short method. So how we can make it innovative and how we can expedite this discovery. So for this, this is very, that's why we are saying this is very multidisciplinary area. Translational research, not like the basic science, not the biologist, not chemist, or not the only engineer people they can do. Everyone should involve. So our course also something like this. Means I am teaching you some biosensor, but everybody should take part here, like basic scientist, biologist, chemist, everything. So this is the two pillar for the translation research. So I mean, for the innovation of the some practice and research, the two main pillars are translation research and implementation. And after the translation, we can implement. So the best things is the collaboration. So we have to collaborate with basic sciences, with biologists, chemists, engineer. Everybody should involve. Then only we can bring this kind of the innovation to the market. So that you should know. Now, I'll show you some important point. I mean, translation research included few phases like T1, T2, T3, and T4. I mean, they have some steps. In the T1 steps, we are developing. Then T2 steps, slowly we are entering to the human trial. And finally, we have to conclude it. Yes, your innovation can be made for mankind and can be useful for this disease diagnosis, for therapy, or for like the biosensing device. Yes, you can go for this one final test. So there is some phases. So I just put this video here for your understanding and for your future reference also, because because I know as you took this class is very multidisciplinary. You should understand the why it is multidisciplinary. And also this research is saying that all together means there is so many phases and we have to face these challenges. So that's why there is so many innovation in our lab. If you see everywhere, we are publishing so many papers, so many journal papers. But if you see market, how many devices there, right? This kind of questions should come in your mind. Guys, is it true?
Yes, sir.
It's very true, right? So we have so many. If you search in Google, there is so many, almost million publication you will get. But we have only few devices in the market. So why? Because of this kind of phases we have to face. Then only we'll get the approval, FDA approval, or maybe see. We have so many in India and abroad also, some authority who are giving the approval. Without approval, you can't bring the device in the market. And who are thinking from now making a startup or something like this, you should know this all the pages because you are planning something bring to the market. So you should get the approval for this. Let's see this video, enjoy it. Then I will take you to the another topic. Let's see what is this.
Opening a pill bottle and reaching for a medication is easy to take for granted.
Is it audible, guys? Everyone?
Yes, sir.
Okay.
Yes.
Okay, let's start.
Opening a pill bottle and reaching for a medication is easy to take for granted. This simple act is made possible by the work of clinical investigators. But how do treatments such as drugs, devices, and diagnostics develop from an experiment on a lab bench to a product that improves patient health? The process of bringing a discovery from bench to bedside is called clinical and translational research. Clinical and translational research can be divided into 4 phases or T domains. Each has its respective characteristics, but many researchers believe that their work overlaps into multiple domains. The phases of the T domains are different than the phases of clinical trials, but they follow a similar path. Scientists spend years in laboratories testing a new treatment using animal models. This is sometimes referred to as T0. If the treatment shows promise, researchers will give the drug to humans for the first time. This step is the first phase of clinical and translational research, called T1 research. In this phase, researchers seek to evaluate the safety profile of the drug. To do this, they give the drug to a small group of healthy volunteers to see how the body reacts to it. This research is often conducted through phase 1 and some phase 2 clinical trials. To see if the drug is effective, T2 researchers give the new treatment to patients who have the disease researchers are interested in. T2 studies are bigger than T1, with hundreds of people at multiple medical centers receiving the new experimental treatment. These larger studies make up phase 2 and 3 clinical trials. The results from these studies help to establish guidelines for how to use the drug in the future. If the new treatment is effective, it receives FDA approval. Doctors can now prescribe the drug in practice, and we have moved into the T3 domain. T3 researchers use the guidelines developed in T2 to teach doctors how to administer the drug in hospitals and medical centers. At this point, the new drug is used on thousands of people. Because other drugs may treat the same disease, T3 researchers compare the cost and effectiveness of the new treatment to treatments that already exist in routine practice. Researchers also start phase 4 clinical trials to continue to watch patients who take the drug to make sure there aren't any negative side effects. The new drug is safe, effective, has been approved, and is being widely prescribed. In the T4 domain, scientists study how the new treatment affects global public health. Researchers continue the phase 4 clinical trials that they started in T3 and study thousands of people all over the world to see how the drug is working. T4 researchers often observe whole communities to try to figure out what may be causing the disease in the first place. What researchers learn in T4 studies can often even change public policy. The pill has finally made it into mainstream treatment, but the path to get here was not easy. The process of developing a new intervention takes an average of 10 to 15 years and costs at least $1 billion US. It's hard to believe that only a small fraction of interventions make it through all 4 domains. And most new treatments don't follow the process smoothly in one direction. Often discoveries made in one domain may necessitate that researchers backtrack into earlier phases. Although the road is long, clinical and translational research can have enormous impact on human health.
Okay, so you just understand that it takes us a long time to bring such kind of the new innovations in the market, right? So anyway, that's why I'm saying we are trying to develop some new concept in the lab, and then we're trying to overcome, bring this kind of device or innovations, and for human, we're trying to see if there's any side effect or not. If there's too much side effect, then we have to go back to the lab again, and you have to solve the problem. Then again come to the human trial, and let's get the approval from like FDA or such kind of approval you have to get. Then only your device you can commercialize easily. Now let's see. I will show you some. Now slowly I will enter to the nanobiotechnology. Right? I mean, this can help you to accelerate this kind of process, and easily how you can bring this kind of your innovation to the market. First is the nanotechnology. It is very much innovative technology. So we can easily integrate your approaches for different kind of treatment, different kind of drug delivery, and their efficacy you can check, right? So this is very much innovative, and we can develop such kind of very small nano machine, kind of very new drug delivery kind of agent you can develop, and you can deliver your drug to the particular area that you need to deliver your drug. You can check their efficacy and their effect, and then you can take the decision, yes, how effective your drug. Also same thing like some device, some bio device also you can make, and you can use this kind of technology, nanotechnology, and you can check the effect. So nanotechnology basically help you to bring your innovation readily to the market. So I will come slowly to the nanotechnology now. What is the nanotechnology? So now make the conclusions. Commercialization part. What is the commercializations of the device? Because that you should know, because I'm just telling you the fundamental things of the commercialization, that what kind of problem you may face and you have to overcome. So that I already showed you last time that we are trying to develop some integrated kind of chip device for biosensor, and if you can design something like this, very small, because it can be user friendly, you can bring to the near the patient, right? So you can see here like this, like a SIM card base, very small microfluidic base, so that small amount of chemical consumption required, and it is sufficient for signal amplification, and we can detect the disease. Or the smartphone integrated things, so that you can see that people near the patient they can use. We don't need basically bulky instrument, expensive and sophisticated instrument. You don't need. So if you can make this kind of very simple things, you can easily commercialize. So this is some criteria for your reference. I think there is, you can see one example, like a very small printed chip. On this chip we are using some paper. On that paper we can draw, we can keep our all the reagent. Maybe we need some nanomaterial, some biological some compound like antibody, some reagent you can keep in the dry form, like I told last class also. If you can make like similar kind of device, so it can be very much helpful for end user to use, like a glucometer case, like a chip, small strip you have seen, just drop the blood and it will come to the sensor surface. Sensor surface already coated with the antibody. So your target molecule will bind and you will check the electrical signal changes after and before. You can easily predict that if there are any cancer marker or any other marker present or not. Okay, let's I'll come to the technology point slowly. I will enter now. Before that, I will show you some commercialization part. Let's conclude this area. What is how you can easily commercialize for your idea? How? So I made this a very small video. This is very not so big, a short video. Let's see here that for kind of the commercialization case, this is very interesting part. And whoever thinking for your future that we want to make something like device and you want to make your career there, you may think this kind of the point, all the points is equally important and useful for you. Let's see this video for you.
Fast Forward Medical Innovation at the University of Michigan strives to help biomedical researchers accelerate innovation from idea to impact. It takes commercialization education, translational research funding, and business development support. A simplified version of the product development pathway includes discovery and ideation, invention and prototyping, preclinical testing, clinical testing, regulatory decision, product launch, and post-market monitoring. Although this appears to be a linear pathway, it is often circular and confusing because of other non-technical considerations, including intellectual property, regulatory assessment, and reimbursement and monetization. Let's build a product development roadmap from concept.
So one thing you should keep in mind while you are developing some new idea. So we have to go for the intellectual IPR, intellectual property. You have to protect your idea so that others, they cannot use, so that before your development, maybe someone else can bring this device in the market. Because sometimes you are developing some idea, somehow maybe if one frankly speaking, because this is very true, if somehow some other company, they know your idea, they can develop. I think this point also should come in your mind. So what you have to do then that's why whenever you feel that yes, this technology is very much relevant for device development, go for intellectual property and protect your idea, and then only you can publish. Yes, your idea already protected. Now slowly you can go to the end step. No need to wait for the long way. Then sometimes you can publish your work after some development, some preliminary work also you can publish. But before that, you have to protect your idea. Is it clear, students?
Yes,
So
sir.
is it?
Yes.
Yes. So this is very important who are thinking out some new innovation, because see, I'm just going to the higher level now. Yes, are you asking any question to me?
Sir, it is an institution where we have to protect, like we have to submit our idea and then we have to protect.
Yes, very good question. See, I will come to your point. So first thing that I'm saying, that whenever you will discover any new idea, you have to go for the protection, like intellectual property, IPR section. Generally, each institute, the R&D section, they are, R&D, they are doing so many innovation. Each institute should have the IPR section. our IIT Kharagpur, we have the IPR section. Okay, like this, all IIT they have the IPR section. Means intellectual property rights. So you can make these things. So how you will go? You have to file. You will be provided an application form. You have to put there all the information with your innovation. Then IPR section, they will ask for a presentation and they will check. And if it is really they find that yes, it is good, it is fine, and everything proved, then you will get your the protection number, and based on that protection number, then you can publish that your idea now protected. So only if someone want to share this kind of idea, they should have to take the permission from you first. Then only they can share your idea. So it is because you are the owner for this. Our IIT Kharagpur has this section, this department. Okay, each institute they have actually. I know while you will be here and who are thinking about some future like this, so you can come here. I can maybe guide you. While you will be at IIT Kharagpur, I can show you also the regulation. Also directly you can contact. You can search IPR of IIT Kharagpur. You can check. So there is you can protect your idea. So this is very important point. Okay, let's now go to the next.
Development through product launch for a medical device. The time from concept to FDA clearance to market for a typical Class 2 medical device can take many years and can cost millions of dollars. So the roadmap needs to be solid before you begin. Here's the plan. Make sure it's needed and that no one else is already doing it. Build a prototype and test it to make sure it works. Find someone to buy it or pay for it. You have an idea that was inspired from a problem or unmet need. Now it's important to clearly define what that problem is, what's causing it, and what happens if it's not resolved. Next, figure out who else is trying to solve the problem. What is the current standard of care? Are there past failed solutions? The best way to find out if your idea is viable is to ask people for customer discovery. Test what you think the problem is and if your idea will solve it by asking potential customers. Customers are clinicians, patients, health insurance providers, regulatory bodies, hospital administration, etc. What is the value that your product adds to your customer? Remember not to sell your idea, but to ask open-ended questions to find out if the problem that you think is a problem is actually a problem that is significant enough for your customer to change what they're currently doing. From the beginning, make sure that you are developing a diagnostic or device technology that has clinical utility and adds value. Let's assume your customer discovery process was positive and validates your hypothesis that your proposed medical device or diagnostic creates value for your stakeholders. Now, let's prototype. It's important to think ahead to the final product that will be used for patients. Here are a few considerations. Be mindful that not all technology developed in the academic setting can be made easily using mass manufacturing techniques. Using FDA-approved materials and/or FDA-approved processes simplify how your product gets regulated in terms of quality control, safety, efficacy, etc. Incorporate continued customer discovery to assess form factors or how your user interacts with the device, its features, and functionalities. Once you have made some progress on your prototype, start to think about your intellectual property or IP. IP protection is usually accomplished through patents. The design and function of your technology must be useful, novel, and not obvious to be patentable. Additionally, be careful not to publicly disclose your technology before it is protected, which means detailing your technology in a public setting, such as a presentation, a dissertation, or other publications. If you are part of an academic institution or health.
So you understand this point, right? So whenever if you want to think that you want to commercialize any idea, this thing, you can't disclose these things before the IPR protection. Nowhere you can publish. You can't discuss with your colleague. This should keep in your mind.
The best way to get IP protection is through your institution's technology transfer office. It's important to contact tech transfer early and often throughout your design and development process. Initiate the conversation by filing an invention disclosure as the first record of invention. Your tech transfer office will work with you to determine a meaningful and useful method of technology protection. At this point, you have a prototype and have talked to your tech transfer office about IP. Now it's time to decide how to test it. The FDA will define the testing plan. The overall timeline and cost to market is varied depending on the complexity of the device and risk to the patient. The FDA allows marketing of devices that are safe and effective and separates medical devices into 3 classes. Class 1 devices pose the least risk to the patient and frequently only require a registration as manufacturer of record with the FDA. Think elastic bandages or dental floss. Class 2 devices pose a moderate amount of risk and most often require a pre-market clearance from the FDA before they are sold. Class 2 devices are the most common and include things like pregnancy tests, infusion pumps, and monitoring devices. Class 3 devices pose the most risk to patients, are generally life-sustaining or implantable, and are subject to the most rigor by the FDA, requiring clinical trials for approval. Think implantable pacemakers and breast implants. As you move from Class 1 to Class 3, the time and cost from initial development to market increases because the amount of evidence required to demonstrate safety and efficacy increases. Let's focus on Class 2 devices, the most common, with a typical 510(k) FDA clearance process. The foundations are intended use, what the device is doing, and indications for use, who this is or is not being used on. The main goal of the 510(k) submission is to demonstrate substantial equivalence, which means that your device is the same as or similar to another approved product in its intended use and the technological method used to accomplish that intended use. For example, a screwdriver and a power drill have the same intended use, but the method by which they work is different. Comparative products in both of these criteria are called predicate devices. When you've identified predicate devices, there are 510(k) summary documents publicly available through the FDA website. You can use these to see how similar devices gain 510(k) clearance to aid in developing your testing plan. If you cannot demonstrate substantial equivalence to a predicate product, but believe that your device is just as safe and effective as similar devices, a de novo 510(k) submission may be appropriate. With proper guidance, it's always a good idea to have a pre-submission meeting with the FDA to confirm the appropriate path to market. Based on your FDA regulatory strategy, let's develop a testing plan. Typically, testing plans involve benchtop testing, preclinical testing, and clinical testing. Throughout this process, your prototype may evolve based on test results. Now that it's been tested and you have a solid prototype, let's consider how the final product will be paid for. It is important to think now about the monetization plan by understanding who your paying customer is. Is it direct to consumer? Is it payers through reimbursement? Is it hospital systems and procurement? Each of these strategies are critical to consider for medical devices and diagnostics. Now that you have assessed the critical components, what is the path to market? Here are 2 main commercialization strategies: licensing your technology to an existing company or forming a startup around your technology. The decisions made throughout this roadmap, as well as industry trends, will most likely determine your path to market. In this high-level overview, we covered the typical path to market and considerations along the way. If you're interested in learning more, check out Fast Forward.
Okay, so this is the story of this, but slowly while you will go to the final stage, then you require, but you should know the story, right? How you will launch your product. Okay, that's short. Now let's come to our nanotechnology. Slowly we will enter now development process. Now I will teach you how we will develop the device. Now one by one, from fundamental things I will teach you. Let's come the nanomaterials for healthcare. So for the development of nanobiosensor, we have to think about the different kind of nanomaterial. They may help you to generate high signal to background ratio. So as I mentioned before, that why we need high signal to background ratio, because we want to make our device very sensitive so that we can detect the disease at the very low level, right? So let's go. Now check the nanomaterial that can be useful for sensor device. So what is the nanomaterials? So is the nano dimension size within 1 to 100 nanometer, and they can be useful because its property, nanomaterial property is something different than the bulk property because of the nano size, and due to the surface energy, their activity also different. Due to the small size, what happened? The most of their, you can check. So you can see that due to the small size, their surface area means if it is like the bulk material, and if you put the nanomaterial bulk material, if you check the total surface area, if it is A1, and if you go to the nanomaterial, I mean like this. So if you check the surface area, total surface area, if it is the A2, so this A2 is very, very high than the A1, because you can see here. So first it is just flat, but when you put the nanomaterial, then it is like this, right? It's like this. So you can see the total effective area is like this, right? So due to this, so one important parameter, you can immobilize so many biomolecule like antibody, right? So if it is like a bulk material, then you will get only like this, you can immobilize. But if your surface coated nanomaterial like this, then you can put here, like within this area, you can put many antibodies, right? So this is the things while we are using the nanomaterial on the sensor surface. This is one part, because you will get the high surface area, and another part is the conductivity. Conductivity. What is this? So if you want to use some nanomaterial like gold nanomaterial or some metal-based nanomaterial, even there is some carbon-based material, then electrical conductivity transfer rate, I mean through the transducer, will be much higher. So you will get the higher signal compared to without nanomaterial. So this is another property. That's why we are trying to use some nanoparticle on the surface for development of the sensor. While you develop sensor surface, that's why we have to check. We have to optimize which nanomaterial is better for this sensing. And we may use some carbon, we may use some metal based. Let's check. There is different kind of nanomaterial available here. You can see like different kind of shape and size also can influence your sensitivity of the sensor. See, sometimes spherical size, sometimes the the rod kind of shape. So their properties differ. So like some core cell type, inside one material, outside another material. So it's like nano prism. So their shape different. Due to the different shape, their property also changes. And the increasing the surface area per unit mass also change the sensitivity of the sensor. But sometimes we don't know which one we will use. Is this one, or that one? So we have to check which one better. We have to optimize. So we have to make a sensor surface, and we have to immobilize them and check it which one better. We have to optimize. So we have to optimize the sensor surface, which one better. So sometimes nanocell or core cell, they may have some important property for electrochemical behavior. The electrical conductivity also may change. You may get higher signal to background ratio. That you have to also may optimize. So this is just a picture of the different sizes of different molecules. You can see I will share this file with you. And I think I already shared few files today morning, a few papers and few books, also slides. Have you checked this one? Did you get all the files?
Yes. Yes, sir.
Yes.
Yes, sir.
You can go through those files, and some books also I share, and some books also myself I also edited, I also published. You can check. It's a very simple book. You can go through them slowly, one by one, and if you have any question, you can ask me also. Okay, now come to the nanomaterials. There is different kind of nanomaterials can be useful for development of the sensor surface. One can be the carbon-based, carbon allotrope-based, like you have graphene oxide, or reduced graphene oxide, or carbon nanotube. So different kind of carbon-based. You can see here this one. This is the carbon-based. You can see their microscopic image. They looks like this. due to their high surface area. You can see the surface area. If you drop, if the sensor, if you use some graphene oxide, then you will get basically this kind of surface. This kind of surface will help to immobilize more biological molecules. This type of nanomaterial will help high conductivity, and because of the high conductivity, your electron transfer rate will be increased. You will get the higher signal. Also, there is some inorganic nanomaterials. Inorganic nanomaterials, that is metallic or some kind of non-metallic, like metallic means like gold. Gold we have mentioned AU, right? Gold nanoparticles we can use. We can use silver nanoparticles. And also non-metallic like silicon dioxide, Si2 nanomaterial. And sometimes this we can use. Also, we have other kind of nanoparticle like organic nanomaterials. Organic nanomaterials means some conductive polymeric nanomaterials. So maybe we can synthesize like amine and COOH group, right? So we can polymerize them. Some organic molecule we can polymerize. Due to their polymeric backbone, their conductivity also may change. You can immobilize this kind of polymer on sensor surface, and they may help you to capture the antibody on the surface. They may help you increase the conductivity. So they can change. So actually, the huge scope you have, you can explore different kind of nanomaterial, different kind of organic molecule, different kind of inorganic molecules. You can explore which is better for this sensor. So now, I'm giving you the general idea. So you can now try basically this kind of things while we'll go for your some research purpose. Which nanomaterial is the better for your sensor? Maybe sometimes gold nanoparticle is good, sometimes it may not good, because gold nanoparticle case one problem may come because it may show some interference effect, some sensor case, because it's highly conductive. If it is too much conductive, in that case some like background current also it may generate, right? So in that case you can go some little bit lower conductive material. Like this you can choose. So you have to optimize which one giving you best signal to background ratio. If in your sensor that all the metallic nanoparticles showing high background, then you have to go some non-metal base like silicon dioxide or some polymer base you can go, or maybe some carbon base. So we have to check which one is better for your sensor. Here is some picture, some carbon based, like it can be like seed, like ball kind, spherical type. It can be seed kind, or can be tube, maybe multi-tube type, or maybe the single tube type, a single wall or multi-wall kind or nanomaterial it can be. So this kind of carbon-based material can be useful for biosensor development. You can see this is the kind of tube, but a spherical tube. They say so antibody can be easily immobilized here, and its electric conductivity also much higher if you use this one. You can check as in the present of this and absent of this, and you have to optimize which one is the best one. So here actually now come to a real example of the biosensor. This is one real example. You can see AU means this is the gold nanoparticle. This is gold nanoparticle. So this nanoparticle actually, you can use the gold nanoparticle on the surface. Here also you can use. Means some general idea I'm giving. You can use the gold nano on the surface. Why you use? Because we increase the conductivity, or maybe you can increase the surface area. Also, you can use the nanoparticle to conjugate with some biological molecule like antibody. So as I said, this is the secondary antibody that can be conjugated with some tag, right? Suppose, see, this one is a target that you are going to detect, right? This is target. So in this case, target is PSA, means prostate specific antigen, or mouse IgG. So just name, just leave it. Leave the mouse IgG. That's PSA you are going to detect, right? Prostate specific antigen. This cancer marker you are going to detect. So if cancer marker present, then it will bind with the primary antibody that is coated on the surface. Don't think this one, that I will let you know later. So somehow you immobilize primary antibody on the surface. So how you can immobilize primary antibody, that I will teach you today. Today itself I will teach you. So somehow your antibody already coated on the surface. Now after the washing, you just drop your sample. If your sample contain your target, then it will bind. It will conjugate with primary antibody. It will bind with primary antibody. This is antibody 1, primary antibody. Now you have to wash it, right? Now if your surface present PSA, prostate specific antigen, your target, then as I mentioned last class, that your secondary antibody also will bind, right? So, because secondary, they have also some epitope where they can bind it. So if I go, I just erase here. I can just erase this one. Okay, now see. So your secondary antibody now already on the surface, right? So your secondary antibody actually conjugated with some gold nanoparticle. So this nanoparticle now will act as an amplifier. How? See, this is kind of a signal, right? Because your target present. So can you remember last time? Let's repeat again one time. So if your surface, when you are adding your sample, if your sample does not contain a target, then when you will add some chemical or some solution, you have to add, right, to get the signal. So if there is no target, and while you are adding some chemical, so what kind of chemical you have to add? So in this case, you are adding this NP, nitrophenol. You are adding from outside nitrophenol. Okay. So this nitrophenol will react with this gold nanoparticle with the help of one reducing agent, sodium borohydride. So basically from outside, you have to add some extra chemical. In this case, nitrophenol and sodium borohydride. So these two extra chemical you are adding. So they will basically react with this gold nanoparticle and will form AP. AP means aminophenol. And this aminophenol now will react with ferrocin plus. And ferrocin plus, this is kind of a mediator or kind of iron compound that is immobilized on the sensor surface. So with reacting with the ferrocin plus, this aminophenol will form a quinonimine. So this quinonimine again will react with sodium borohydride that already in the solution. It will form again aminophenol. Then again this aminophenol will react with FC plus and will form the quinonimine. And then FC plus will be converted to the FC0. Now see, FC plus means it is plus. It means it is the higher oxidation state. Higher oxidation state. FC0, it means lower oxidation state. Oxidation state. Now please let's come to this point. I think you already know what is oxidation state. Let's teach you. Like Fe, if it form Fe plus. Fe plus means it will release one electron. If iron release one electron and form the Fe plus, this process called oxidation, right? So oxidation means releasing the electron. And reductions means if it receive one electron, means plus E. If it receive one electron and come to the Fe0, then this process is called reduction, right? Do you know this, students?
Yes, sir.
So this process is called reduction, right? Okay, so now what we are doing, your surface already contain Fc plus, means higher oxidation state. Then this Fc plus will be converted to the Fc0, lower oxidation form, and your aminophenol automatically will be converted to the quinonimine. So Fc plus will form Fc0 and aminophenol will convert to quinonimine, QI. This is this form. And this form again will react with sodium borohydride and this. So this is basically a cyclic reaction. So why we need this cyclic reaction? Because of this cyclic reaction, your electron transfer rate from nitrophenol to this surface that electron is transferring, that transfer rate will be enhanced. Okay, so if your surface contain only nitrophenol and there is no gold nanoparticle, then see, if there is only nitrophenol, but you can see here aminophenol can react with the Fc plus, not nitrophenol. And nitrophenol and sodium borohydride, they can react and they can form the aminophenol AP if there is gold nanoparticle present, right? So if there is no gold nanoparticle, then you can see there is no aminophenol basically. So if there is no aminophenol, then Fc plus cannot be converted to the Fc0. So let's remove all these things, then I will repeat again. Let's remove. So I think basic part I already described. So I will repeat again. I think it should be clear, because this is the basic reaction mechanism, how to amplify the signal and how we will get high signal to background ratio in an electrochemical biosensor. I will repeat again this slide because you should make it clear first. Okay. See, so this is your biosensor, right? This means primary antibody coated biosensor. You will be provided. So this is your sensor surface. Now you are adding your target solution, right? Your sample. So sample you are adding on the sensor surface. That may contain your cancer marker, prostate specific antigen, right? So if your sample contain prostate specific marker, then it will bind on this primary antibody. Okay. Now, if as the marker present, I mean target cancer marker present, then while you will add your gold nanoparticle conjugated secondary antibody, that will bind. But if your sample is 0 concentration of your biomarker, I mean target, then this secondary antibody will not bind. Right, students, is it clear? If it is 0, then this nanoparticle will not bind. So basically you will get 0 gold nanoparticle on your surface. But if your sample content a cancer marker, then you will get gold nanoparticle on your surface. Is it clear?
Yes, sir.
Yes, sir.
Let's now come to the next step. So why we need the gold nanoparticle on your sensor surface? So basically gold nanoparticle helping you to generate the signal. How? Because in this biosensor, you are adding some extra chemical to generate the signal. What is the extra chemical? It is the NP, nitrophenol, and NaBH4, sodium borohydride. So this is your starting material that you have to add while you will generate the signal. So how will you generate? So as your sensor surface already contain gold nanoparticle, then your NP, that is nitrophenol, will be converted to the aminophenol. So this aminophenol only will be on your sensor surface if there is gold nanoparticle. So basically, if your sample is 0 concentration of prostate specific cancer, then you cannot generate this aminophenol. Clear? You can't generate this aminophenol because there is no gold nanoparticle. Now, as you have prostate specific marker present in your sample, so you are generating an aminophenol, and your sensor surface already coated with some iron mediator. This is called mediator. Why they are mediator? Because they are actually be unchanged always. They are converting like Fc plus to Fc, Fc to Fc plus, like this. So they will be converted like this, Fc plus to Fc, Fc to this. Means they're like kind of catalyst. So they're actually helping, they're mediating the electron transfer process from nitrophenol to the sensor surface. So electron actually transferring from nitrophenol to the sensor surface, right? So if you don't have target, then only nitrophenol cannot transfer electron because they're not that much active. So they're less active. What is less active? Less, you can say electroactive. They are less electroactive. So as I told you last time that you have to choose your starting material, your reagent, something like this, so that they will not react very fast on your sensor surface. See, I choose here nitrophenol. And already your surface present ferrocene plus. And this ferrocene plus and this nitrophenol, they should not react very fast. You can't avoid, but you may get very low amount current, that is called background current. So your background current will be responsible because of Fc plus, nitrophenol, and sodium borohydride. Because your starting reagent, which one? Which one is your starting reagent? Nitrophenol plus sodium borohydride, right?
So, yes, sir.
So these two should not react, right? If they react, then nitrophenol will be converted to aminophenol, right? So these should not react. So you have to choose. I mean, you can definitely change nitrophenol. Maybe you can go something else. So what is the something else? That something else should not react with the sodium borohydride. That should not react with this Fc plus. Why? If they react, then your background current will be very high. Because anyway, we are going to generate high signal to background ratio. If background current high, then you will not get very high signal background ratio, right? Okay. So this nitrophenol, sodium borohydride, and Fc plus, they are not reacting fast. That is clear. Now, as your surface contain now gold nanoparticle, and we already checked, because in the presence of gold nanoparticle, nitrophenol and sodium borohydride, they will react and they will form aminophenol. Now, if your solution contain aminophenol, they immediately will start reaction with Fc plus. So nitrophenol was not reacting, but aminophenol can react very fast. So then your Fc plus will be immediately converted to Fc0, and aminophenol will be converted to a quinolimine. So it is kind of the one is oxidation, one is reduction. This oxidation reduction can take place simultaneously. So you can see here aminophenol itself oxidizing, right? And it formed the quinolimine, but it is reducing Fc0 to Fc plus to Fc0. See, they are giving the electron. They are giving electron here. That's why Fc plus convert to the Fc0, right? And itself oxidizing because it released the electron. That's why it formed the oxidized form quinolimine, right? Now it is already oxidized form, so it can react with the reducing agent, like sodium borohydride is a reducing agent. This is reducing agent. So it will react with sodium borohydride, itself will convert to again aminophenol. So now it reduced again. Now it can oxidize to quinolimine, but it can reduce to Fc plus to Fc0. So this step is the oxidation step, and this step is the reduction step. Okay. So now you can ask me why you want Fc0 always. Why you use the Fc plus? I use Fc plus because I want to generate Fc0 so that I can apply here a oxidation potential from outside on the sensor surface. So on sensor surface, I will apply like plus 0.3 volt. So some potential. So this is called oxidation potential. So some oxidation potential will apply from outside so that it can oxidize the F0 to Fc plus, and it will release the electron. So, but if you apply 0.3 volt on the Fc plus, you cannot get the high current because it is already oxidized, right? It is already Fc plus. You cannot get Fc plus to Fc2 plus because it is already oxidized form. it is not thermodynamically favorable. Fc plus to Fc double plus not favorable. That's why you will get the low current. But Fc0 to Fc plus is thermodynamically favorable. You will get the high current. That's why we are generating Fc0 always on sensor surface. After generation of Fc0, you can apply some potential from outside. Suppose 0.3 volt potential you will apply. Then it will release the electron. That electron will measure. You will get the high signal then. So basically, this oxidation reduction is basically help to transfer the electron from the nitrophenol to the sensor surface is very fast. It makes the transfer process faster. And you will get the high signal. And your background is low. Okay, so this is the reaction mechanism on a sensor surface. So slowly I will come to the now sensor modification. Today I just showed you one reaction mechanism on a sensor surface. Just make it clear.
So I have a doubt.
Sir,
Yes, I will take the question one by one. So I think it's better. Let's remove these things, right? All the things. Yes, please ask the question.
So I couldn't
sir,
understand the particular.
How?
Sir, I couldn't understand the particular role of AU nanoparticles. Like it is acting. Is it acting as a catalyst or like something else?
Okay. So here actually yes. This is the nanoparticle here acts as a electrocatalyst. This is kind of the electrocatalyst. Yes. Only if gold nanoparticle present, then only it convert this one. This is kind of the catalyst here. Okay. So this catalyst actually basically make your sensor surface very specific, right? It's make it specific. So if nanoparticle present, then only this reaction start. Otherwise, there is no reaction. So this is a electrocatalyst. Okay, next question.
Sir, how long QI converts into AP and AP into QI?
Okay, how long it will be converted? So this is the starting material. Generally, this one we will use some little bit higher concentration. Higher concentration we are using around 5 millimolar, something like this. So that's why generally for the whole reaction we may have to wait for 10 minutes. So we have to optimize. That's why our reaction condition. So we cannot use very low concentration. At least some 5 millimolar, something like this we have to use, so that at least we may get 10 minutes. So because it is consuming, right? So they are not changing. But sodium borate is slowly consuming. Nitrophenol slowly consuming, right? So whatever we are adding from the outside, that is called the substrate solution. So we have to use some high concentration of substrate concentration so that they should not be consumed within 10 minutes. Clear?
So, sir, can we quantify or analyze what we are detecting with this method?
Yes, we can quantify. How we can quantify? Based on this primary antibody, right? So we are developing a biosensor. So in this case, prostate specific antigen specific biosensor we are developing, right? Right?
Yes.
What is your name? I can't see because I opened the slide. Huh?
Shiva Tomar, sir.
Shiva Kumar. Okay, Shiva. So you are going to develop a biosensor for prostate specific antigen, right? So based on this, you are immobilizing a primary antibody. So your primary antibody will be specific for prostate specific antigen. At the very beginning, I mean, first class, I told you that our biosensor is very specific. They can detect only your prostate specific antigen, not others. So although your sample may contain other cancer marker, other thing, or other interfering species, they will not bind. Only prostate specific antigen will bind. Because you are using only prostate specific antigen specific primary antibody. So that's why you can quantify only prostate specific antigen. Clear? Shiva?
Sir, so in this way, just suppose only one analyte, I mean, prostate specific antigen is present, then it will convert into AP, and then it will recycle through QI, and it will generate current. So in such a way, we will get high current, but analyte is only one.
Yes. So in this case, analyte one. This is called a single detection. Because you are going to detect only single. But if you want to detect like cardiac biomarker, cardiac troponin I, do you know these things? What is your background, Shiva?
Chemistry, botany, zoology.
Okay, fine, no problem. So cardiac troponin I is the another biomarker for cancer, cause heart disease. So if you want to detect these two things together, right?
Yes.
So what you will do? Then you have to form a multiplex sensor surface. Okay, you have to form a multiplex sensor surface. So one surface, you have to immobilize the primary antibody that is specific for PSA, and another sensor surface, you have to immobilize a primary antibody that is specific for cardiac troponin I. And these both will be in the same solution. Okay, and this will be connected with the reference electrode, counter electrode, and all the setup will be connected to your electronic device that is called potentiostat. With this electronic device, you can control the potential, you can measure the current. Okay, now if your sample contain both, then both antigen can bind. But if your sample contain only PSA, not troponin I, then this will not bind. Then you will get only signal from here, you will not get any signal from here, you will get 0 here, but you will get some signal from here. And from here you can say your sample content PSA but not troponin I. Clear?
Yes, sir. So we will detect only, right, sir? I mean, rather than quantifying how much antigen is present.
Yes. Now come to the second question here: how much is present, right? For that, say you have to make a calibration curve. That anyway, I will come next after few class, not today. But as you asked, I will just show you what you will do. So suppose this is just 1 nanogram present in your first sample, right? You got a current here. So this is like current versus concentration. You have to make a graph. Current versus concentration. So suppose you got like 1 nanogram this much. Then you increase concentration like 10 nanogram, right? So because as you increase 10 nanogram, there is so many antigen present, right? So more nanoparticle will be there, correct? Correct, Shiva?
Yes, sir.
Correct?
Yes, sir. Yes.
So more nanoparticle means more nitrophenol will be converted to aminophenol, correct? So
Yes,
more aminophenol means more FC plus will be converted to FC0, right? So now,
yes,
as on your surface so many FC0, then you will get the very high current, correct?
yes, sir.
Then you will get high current. So you just put here in the calibration curve, top. See, so current versus concentration, you are making a curve. So 1 nano, 10 nano, increase 100 nano, increase 1 microgram, right? Okay. So then you just make it like this curve. So before going to the unknown concentration, in your hand, you will have a calibration.
Sir, I have a question.
Okay, please ask.
Sir, I have a question.
Yes.
Sir, you are showing that the NP is converted into AP in presence of sodium borotetrahydrides.
Yes.
So, sir, there is an increment in 2 hydrogen molecule.
There.
there is increasement in the 2 hydrogen molecule
Yes.
in this reaction. So
Yes. Continue. I can't get you. I can't hear you.
may I?
No, your voice breaking. I can't hear you. Yes, continue. Please repeat your.
Are you adding one hydrogen molecule extra in this reaction?
Extra hydrogen molecule? No. I'm not adding extra hydrogen molecule. We are adding like see, right? Yes. Continue your question.
Where 2 hydrogen molecule is come from where? And in this reaction, after then AP when converted into QI, then in this reaction 2 hydrogen molecule is one hydrogen molecule is liberated. After liberation, this hydrogen 2 molecule is when QI is converted into the AP, then one hydrogen molecule is added in this added in the reduction process in presence of sodium borotetraidide. But when initial reaction occur, NP is converted into AP. Then in this reaction, I am not understanding where 2 hydrogen molecules came in this reaction.
See, we are adding some little high concentration of sodium borohydride, around 5 millimolar, right? Like Siva asked me that I may answer it, right? 5 millimolar. So this is a high concentration of sodium borohydride. So sodium borohydride will react with this nitrophenol, and excess hydrogen molecule will be released from the solution as a hydrogen molecule. It will release, and they will give you the hydrogen plus basically. For conversion of this, they are giving the hydrogen plus for this reduction, and excess hydrogen will be released as a hydrogen gas. Okay, but suppose you have 5 millimolar hydrogen molecule, and suppose 1 millimolar already consumed because of this conversion, but still you have 4 millimolar, right? This 4 millimolar now will help to this conversion analysis, because you have lots of sodium borohydride. Some hydrogen plus will help you for conversion this, and excess hydrogen ion, they can be released as a hydrogen hydrogen as a molecule, as a gas from the surface also. You will see lots of bubbles also. You can check that there is some bubbles also there. Okay. Okay. Now, okay, fine.
Yes, sir.
Okay, now come to the Siva's questions. Then how we can quantify? Siva is thinking maybe we can check like yes or no. No, it is kind of the quantitative, not qualitative. So our biosensor is the quantitative. We can quantify how much cancer marker present in your sample. So first you have to make a calibration curve. This is a calibration. So how will make calibration? First, from the known sample, you can make current versus concentration curve. So like 1 nano, 10 nano, 100 nano, 1 micro, increase, you will get higher and higher current. You will make this calibration curve. So now in your hand, you have this data. Now what you will do, you have to take an unknown solution, right? Unknown solution. And you don't know how much PSA present there. What you'll do, you just drop it and run your reaction and collect the data. Suppose you are this is 1 nano, this is 10, this is 100, and this is 1000, this is 10 to the power 4. So this is like 1, 10, 100, 1000, 10 to the power 4. So this is current value, and here is the concentration value. In the x-axis concentration, y-axis current value. Suppose you got your current is 95 milliamps, right? You are getting from your unknown sample. Then go here in the calibration curve, you're 95 here, right? Here 95. Then draw a straight line here. Then it cross your calibration curve. Then come again to the straight line to the x-axis. is this much. So it is around, suppose it is 25 nanogram per ml, right? So then you can quantify your unknown sample content 25 nanogram per ml prostate-specific antigen. Is it clear, Shiva?
Yes, sir.
So you can quantify, right? Not like yes and no question. You can get the answer, how much.
So as we see that this reaction is a cyclic reaction, so the like time period for which we allow the reaction to happen will not affect the current output.
Sorry, can you repeat again?
Am I audible?
Yes, you are audible. Please repeat the question.
So I said, as we can see that this reaction is basically a cyclic reaction. So the amount, like the time amount for which we we keep the reaction happening, won't it affect the current output?
No, it affect. See, this is the cyclic reaction, right? And for this conversion, see, that's nitrophenol converting to the aminophenol, then aminophenol reacting with Fc plus, then Fc plus from the Fc0. Then for these productions, you may have to wait for few seconds, at least few seconds. So what generally we are doing, we are waiting for at least a few minutes, like 5 to 10 minutes we are waiting. See, suppose you have in your sample like 10 unit Fc0 on your surface, right? So let's wait so that all the, I mean, suppose if your solution contain like one gold nanoparticle only you have. So all within 10, we have to wait for 5 to 10 minutes. So suppose you are waiting for 10 minutes. So within 10 minutes, what will happen? This gold nanoparticle will convert the NP to the AP, and then Fc plus will form the Fc0. So your reaction will be almost will be somehow, it will be saturated within 10 minutes, and you will get like here 5 unit F0 will be on your surface, because all Fc plus will not be converted, because you are actually on your surface will be very high amount Fc0. Okay, so within 5 to 10 minutes, your reaction almost will be saturated. But you should not wait more than 10 minutes. But if you want to wait for 11 minutes, then within 1 minute also, some Fc plus can be converted with the Fc0. That's why your incubation time will be always constant. When you will make a biosensor, you are incubating something, you have to use your timer. Let's wait 10 minutes means 10 minutes, not more than 10 minutes. So if you wait for more than 10 minutes, then some again Fc plus can be converted. But don't start your reaction immediately within 1 second, because immediately their change is too much. Within 1 second, 2 seconds, 3 seconds, because there's so many reactions going on the surface. It can come to the slowly saturation within 5 minutes, something like this. Not 5 minutes, we don't know when, but if you wait around 5 to 10 minutes, almost all the reaction will be completed. But still, it is not stopped. If you wait like one reaction for 10 minutes, one reaction for 11 minutes, one reaction for 12 minutes, then you will not get the reproducible data. 10 minutes means 10 minutes, always. So you have to stop your reaction, then you have to start the measurement immediately. Clear now?
Yes, sir. So after the saturation happens, then we apply the oxidation potential and measure the current.
Correct. After the 10-minute reaction, then you have to apply 0.3 volt. Then you will immediately it will generate the current that you have to measure, and that current you have to compare with this calibration curve. Okay.
Yes, sir. Thank you, sir.
Thank you. What is your name? I can't see anyone because I just opened my slide. What is your name?
Shazia.
Shadia. Which department you are from?
Sir, Triple M SMST.
Oh, you're from SMST. So what is your specialization?
Sir, Triple M.
No, your bachelor's biology or chemistry or mathematics, which one? I mean, you already passed, you qualified, right? Bachelor degree, right? It is your master degree.
Yes, sir. Yes, sir. Sorry, sir. Actually, my network is.
Okay, your bachelor, which degree?
So biotechnology.
Biotechnology. Okay, fine. So as you're a biotechnology student, so I think you can understand this basically antibody antigen binding, but you know the chemistry also, right? This basic chemistry. Huh? Anyway, so if any students,
Yes,
if you don't understand this all the mechanism, these things, please ask me. I can repeat many times because it is the very first, you are the very beginner in this course, so I can repeat, and at least you should learn the basic thing first. Then only you can slowly understand the whole device development. I can take more question today. No problem. Any question from your side, students? If
Sir,
it's
I
not,
have a I have a question, sir.
okay. Yes, continue. What is your
The
question?
My question is that why we need the FC and FC0, because we can calculate the electron from NP and QI itself, right?
Yes.
AP and QI, we from this reaction we can calculate the electron by
Yes.
using potential. So why the use of FC and FC0 specially?
Very good. Very good questions. What is your name?
Viprodeep, sir. Viprodeep.
Biprati. Hi, Biprati. Very good question. This one. I think I was waiting for this actually. Maybe after a few class, I will teach you. Anyway, as you asked me, I can explain a little bit today. Then slowly you can understand when I will teach you the redox cycling amplification for biosensor. This topic. But as you asked me today, I will let you know why. See, so we are mainly transferring the electron from here to sensor surface, right? This electron is transferring. So anyway, you can see, as I mentioned, this is the mediator, right? Mediator. And this aminophenol also acts as a mediator. This is mediator number 1. You can say this is mediator number 2. So if you see the one cyclic reaction, cycle 1, and here another cycle 2. In the cycle 1, so nitrophenol converted to the aminophenol, if your sensor surface here, then you can apply some potential here. Then this aminophenol can be converted to the quinonimine, right? So if we apply some potential, this aminophenol can be converted to the quinonimine because of potential, right, Vipradeep?
Yes, sir. Yes.
So if you apply some potential, because this is already the reduced form. This is the oxidized form. This is oxidized form, and this is reduced form. So if you want to oxidize by applying some oxidation potential, then it will release the electron. It will form this, and it will be converted again this because of sodium borohydride, right? Then we can get the electron and we can get the signal 1. Okay. Now come to the cycle 2. So if you want to employ another cycle, so how you can employ another cycle? By using another electron mediator that should be in the higher oxidation form. So why we need higher oxidation form? Because this aminophenol already in the reduced form, so it should reduce it. That's why we use the higher oxidation form, or it is called oxidized form. So this one will be reduced, it will form this one, and it will generate another cycle, that is cycle 2. So this cycle 1 and cycle 2, both when we combine, we have seen this electron transfer rate H2. If you see this electron transfer rate H2 signal 2, it's much higher, almost 100 to, you can say, 1,000 times faster than the only H1. So why it is faster? Because another cycle, they're accelerating the transfer process from aminophenol to the electrode surface. It is actually accelerating, or you can say it is kind of the mediator, actually helping electron mediation from, again, aminophenol to surface. So basically this FC plus helping this transfer process much faster. So if you combine again one cycle, cycle 3, you can see then S3 will be almost 10 to the power 5 times faster than the S2. Why? Because this again cycle, it will accelerate this electron release from Fc to the electrode surface. It will be much faster. So I will teach you anyway this cycling process. This is called actually redox cycling amplification. So if you put many cycle, so basically step by step you are accelerating the transfer process and you are amplifying your signal. Clear, Vipradeep?
Yes, sir. Yes, it's clear.
It's very good. You have a good scope to get the high signal. But then one question Vipradeep may come, or someone can ask one questions. Can I use a 5 times, 6 times, 7 cycle? Then we'll get maybe 10 to the power 10, 10 to the power 11 times signal amplification, right? So why not people are doing that much? Why only 2? Why only 1? Sometimes people use only 1, not 2 also. Can you answer this one? I already answered anyway. Vipradeep, can you answer? If I now ask you your question to you, so can I use again cycle 4, cycle 5, cycle 6 like this?
So, sir, it will take many time, and as it's a circular process, so automatically it will converting and returning to its previous phase. So for the time, if we can't collect the electron like for
No, you can collect definitely, because see, it is electronic level, very nano level reaction going on, right? So I don't think that you can't collect. You can collect. So just think.
Sir, maybe it will give more background current.
Yes. Okay, I'm the giving now answer. See, you have to think always signal to background ratio while you are generating many cycle. While you are developing any sensitive biosensor, always you have to consider this signal to background ratio. So if your requirement to achieve like 1 nanogram per ml, right? This is the disease level, and your normal level like 100 picogram. It is 1 nanogram disease level. Why you want to go 1 femtogram per ml? You don't need to go that reach to that much sensitive level, because if you reach 1 nano, then it is enough. If you want to employ here like 5 cycle, 7 cycle, unnecessarily you are making your sensor complicated, because you have to add like 7 to 8 varieties of chemicals, right, Vipradeep?
Yes, sir. Yes.
So that chemical unnecessarily making your sensor expensive, also complicated, because you have to use so many thin membrane to dry this form. Also another problem, just I got some answer, some of you just told me the background current. Unnecessarily they can increase also background. Maybe so selection of this chemical is very not very easy. As I mentioned, this nitrophenol and Fc plus, they should not react. So as you are going like 7 setup, you can see somehow maybe if you go for permutation combination kind of thing, they are not reacting, but this can react. These are not reacting, but this can react. Then see, it will be very much then difficult for you to develop a sensitive biosensor. No, that's why we are not going so many cycles. At least 2 to 3 cycle is enough, because 2 to 3 cycle we can achieve within femtogram level also. Sometimes you can see some publication there, only 1 cycle they use. Why? Because within this cycle they achieve their sensitivity. So then why unnecessarily will make our sensor complicated? I think got your answer, right?
Yes, sir. Yes.
Okay. So please ask me the question, because this is very, very basic part you should learn. Huh? Then only I can teach you the next part. Clear? Can I go next? Okay, let's come next. Now, bioimmobilization. So as I told you, see here I immobilize the primary antibody and the sensor surface. This is the immobilization step. That step now I'm going to teach you. How we'll immobilize the primary antibody. So I can see primary antibody, it can be easily immobilized on the ITO. ITO is the electrode. This is called the transducer is the ITO. This is transducer. So how will immobilize? So we have to use some linker. This is called linker. Some linker. They will help you to bind on the sensor surface. So what kind of linker we can use? Let's teach this one. So bioimmobilization. See, your antibody, that is your bioreceptor. So I am always taking an example of antibody. So it can be anything like your aptamer, it can be something other receptor also. But as for example, I am taking antibody. So antibody may contain some free functional group. It can be some amine group or it can be some carboxylic group. Okay, amine means NH2, carboxylic group means COOH. So all the antibody, they have this kind of functional group. Now, what we will do? We can add some linker with the antibody. They will help to bind on the sensor surface. Okay. So suppose you want to consider the amine group. Let's go to, let's erase this one. So you want to modify your antibody on the sensor surface. So this question may come on your mid-sem exam. How you will modify the antibody on a gold nanoparticle modified surface? Something like this question I may ask. So suppose this is, sorry. So let's, so suppose this is your sensor surface, right? This is your sensor surface. So on your sensor surface, you can functionalize with some functional group. What functional group? Like carboxylic acid. Just one example. Carboxylic acid, you can functionalize. Now one question you can ask, how we can functionalize carboxylic group on the surface? There is some example there. You can use some nanoparticle for this. So like this way, nanoparticle can help you to functionalize on your sensor surface. So to coat a antibody on your surface, you have to functionalize the surface with carboxylic group. So you have to take a nanoparticle like carbon nanotube, some commercially available nanoparticle, you can synthesize also. Some nanotube available. Their surface contain lots of carboxylic group acid. Okay. So you can print on the surface, you can drop cast on the surface, or maybe you can electrochemically also you can coat on the surface. Drop cast means you just drop it on this nanoparticle on the surface, then your surface will be coated with carboxylic group. So by using some carboxylated nanoparticle and by dropping them or by immobilizing them, so you will get carboxylic acid group contains sensor surface. Now, so number one, you can see here, this is number one, COOH. So this is your surface, sensor surface. Okay. So on this surface, you have to add this linker. This is a chemical that is called EDC. The full form of EDC is this. N3-dimethyl laminopropyl, N' ethyl carbodiimide. No need to remember. Just you can tell that EDC and you can check in Google, you will get the full form. So no need to remember the full form. And you will get the structure this. So while we will add the EDC with the carboxylic acid group, so basically they will activate this COOH group and they will link like this. And this group basically help to react with the amine group with the antibody. As I mentioned, the antibody may contain with lots of amine group on their surface. It has so many free amine group. So antibody contain free amine group, that amine group can bind now with this carboxylic group. So in this case, this linker basically help to bind with this amine group. Now it will react with this and it will form this kind of amide bond. 1 and 2, see they already conjugated. So and this one, this group left. So basically this is kind of a link, this is kind of a activation group. This group is the activation group, and this group basically help to bind this carboxylic group and amine group. So you can ask me why we are adding the EDC. If we add like number 1 and number 2, then we can get this form. You may get, but that may take around a month or maybe maybe a few years also. They will not bind because they are thermodynamically they are okay. They can bind. I mean the carboxylic acid group and amine group, they are thermodynamically they can bind, but kinetically they are very, very slow. Right? They can't bind. So for that you have to add this EDC. So this EDC make this reaction much faster, so that 1 and 2 they can bind with the amide bond. So basically then see this primary antibody, it contain amine group, and you are adding here EDC. Because of helping this EDC, this amine group will be on your sensor surface. Clear, students?
Yes.
Sir,
Yes, sir.
what's the cross-link protein?
Tell me.
What is the cross-link protein? I didn't get that reaction.
A cross-link protein. See, cross-link, see, because we link together, right? On number 1, number 2. So instead, see here actually I just mentioned this is the surface, right? So instead of you can put something else also here. Maybe it can be another protein that contain carboxylic group, right? Or it may be sensor surface, can be nanoparticle. I mean, you can cross-link anything by using this amide bond. So this carboxylic group I put. Okay, let's remove everything. Discard it. See, so here I mentioned this carboxylic group on 1. So it can be on sensor surface. It can be on some other biological molecule. It can be on a nanoparticle, anywhere. Then on that surface you can bind this primary amine group that comes from the antibody. You just come back here. Now if I ask you how you can bind a primary antibody on the gold nanoparticle, how you can bind? So your gold nanoparticle surface may contain carboxylic group, right? You can functionalize the surface with some carboxylic group. So how that can be possible? By oxidizing your gold nanoparticle surface or by adding some carboxylic functionalities. Now if your primary antibody that contain the amine group, now add the EDC, then easily your gold nanoparticle can bind with the antibody. This is the cross link. Clear?
Sir, focus.
Yeah, so this is very, very basic part I am teaching, these few classes. So if you have any questions, please ask. Any more questions, students? I think let's come back to you. Long time I am teaching. Yes.
Sir, how long cross-link will occur?
how long this cross-link will occur. That depend on the concentrations, how much you are adding, and you have to incubate it for, yes, you have to check this one. It is not like this within a few minutes it will be completed. You may need some time. Maybe it may take around 30 minutes, it may take 1 hour, it may take 1 day also. That's depend, sorry. So it may take some 1 hour also, so it's depend. Okay, clear?
Yes.
It's depend. So anything else? Any more questions you can ask? Hello?
No, sir.
No, sir.
No,
No questions. Okay, good. I think this is clear, right? Up to this. So I can now let's teach you one more. Where is this? Okay. Let's teach you the one more steps, bioimmobilization steps. Yes. See. Here. Sometimes we use the EDC. Also, we are adding one extra chemical. Just now you asked that, sir, how long we have to wait. We can accelerate this process by using some other chemicals also, like EDC, also NHS. You can see here. Okay, let's put the pointer. See here. So this is the carboxylic acid functionalize your sensor surface, right? So you are adding here EDC cross-linker. So EDC will bind like this, okay? Then you can add another chemical, that is sulfo NHS, another chemical. So then what happened? This linker actually leaving, right? Then primary antibody that contain the amine group, it will bind and it will form this cross-linked product with carboxylic group and amine group cross-linked product. So one can be your sensor surface, can be nanoparticle, anything. Now, if you see, if you add here one more sulfo NHS, then this link can be removed by the sulfo NHS here, but its leaving aptitude is much higher than this. So that's why generally what we are doing, we are mixing the EDC and NHS together to make it much faster and more effective binding. So stable conjugate. To make it more effective binding, what we are doing, we also add the NHS. Okay. So this EDC plus NHS, if we add, so first EDC will bind, then it will be removed by NHS, sulfo NHS, and its leaving aptitude is much faster, so that easily this amine group will bind with one, and you will get this product, cross-link product. So instead of that's why this is our idea. We can add the mixture of EDC and NHS to get this conjugated product. Okay.
Sir.
Yes.
Yes, sir. So I was saying, sir, like if we add the mixture of EDC and sulfo NHS, then why the EDC is binding fast, not the sulfo NHS?
Oh, see, here's EDC. Actually, sulfo NHS can remove this part here. So first, actually, EDC can bind here. Sulfo NHS cannot bind. Sulfo NHS cannot bind fast. Okay. And this one can be easily removed by the sulfo NHS. And sulfo NHS leaving aptitude is much higher.
Sir.
Yes.
Sir, Akhilesh sir, a carboxylic acid when converted into the intermediate O-acyl urea, and after reaction with water, it again converted into carboxylic acid. What is
Yes.
the difference between them?
See, that's why here, so although you are using the EDC, so this water also, there is some competitive reactions with water and also the NaCl. There will be some competitive reactions. So that's why we have to optimize the concentrations of this. Which concentration you have to use, sulfur NHS, so that water should not come. Your sulfur NHS should replace this one and should form this one. So if you don't have sufficient concentration this one and water can easily replace, you will not get your expected product. That's why I showed this one. But this is some, I mean, a side product you may get. This also some unreactive one. That's why you have to optimize the concentration of sulfur NHS also. Maybe you are expecting some conjugate, but your conjugation not formed because your concentration is not that much high. So there is some byproduct possible. That's why you should keep in mind. That's why I put this one. So if there is not that much sulfur NHS present, then you may not get your conjugate one. Clear?
Okay, sir.
Good questions. Keep asking. Okay. Now let's show you another thing here. So I'm teaching you the bioimmobilization, right? So your sensor surface should contain some carboxylic group acid, also maybe some other group. Why always carboxylic acid, right? Maybe we can put some amine group also on the sensor surface. So how we can put your sensor surface amine group? See. So first you can form your sensor surface, some carboxylic group. How we can form carboxylic group? You can use some carbon nanotube that coated, that already functionalized with carboxylic acid, and you can drop it, or you can print it on the surface. Then you will get some carboxylic acid group surface. Basically, you can oxidize also sometimes in sensor surface. You can get the carboxylic acid group. So some carbon-based material you can use that contain lots of carboxylic group. That's the one part. Second part, how you can form amine group on your sensor surface, right? So your primary antibody has so many amine group, and you can modify your sensor surface amine group, and you can link this sensor, your sensor surface contain amine group, and this amine group also together you can link. How? Let's come step by step. So first step, just leave it GMR. This is some other name, so don't remember the GMR. Just think this is a sensor surface. So your sensor surface should be very much cleaned before adding any kind of functional group. Why I am telling you your sensor surface should be clean? Because when you are getting the sensor from any company, maybe some ITO electrode, I mean gold electrode, or maybe carbon electrode, in some electrode plate, it may contain so many impurities. Different kind of impurities, some oil, some other contaminant, right? Because they are fabricating industrially. Maybe some people, they touch the surface, right? So some contaminant may come. They can cover the surface. And that kind of contaminant can make your problem on your sensitivity. You may not get the reproducible result. Clear why we need cleaning the sensor surface before using for biosensor or before functionalizing for biosensor. Okay, clear? Students?
Yes, sir.
Yes, sir.
Yes.
So you have to clean this. You have to clean the sensor surface always before functionalization. Why? Because when we receive from the company or from industry, maybe they use their process. Maybe someone they touched or some contaminant may come. Right? They can hamper. So you have to clean. So there is some procedure, cleaning procedure. So while you clean the surface, then you will get lots of hydroxyl group on the surface. This hydroxyl group make your sensor surface hydrophilic. Your sensor surface will make hydrophilic. Do you know what is hydrophilic, hydrophobic? Anyone?
Yes, sir. Yes, sir.
Can you tell me what is hydrophilic? What is hydrophilic?
Hydrophilic, which react with water or say attract towards water, like polar group, and
Polar.
hydrophobic, which yes, sir.
Yes, hydrophobic means not much attracted with the polar group, right?
Yes.
Oily kind of thing. So generally you have to make your sensor surface, because we are adding so many we are generally dissolving our chemical in water and we are dropping on the surface. Your antibody also we are going to drop on the surface in the buffer sample that contain water. So your surface should be hydrophilic. Okay, so your surface should be hydrophilic, not hydrophobic. So hydrophilic means water can be attracted, right? So your contact angle will be low, right? For the hydrophilic surface, your contact angle will be very, very low. So if one surface is hydrophilic, then if you measure the contact angle, so this is the contact angle, right? So contact angle will be very low. If your surface is hydrophobic, then you will get like this water drop. Your contact angle of the hydrophobic will get very high, right? Students, do you know this?
Yes.
Yes.
Okay. So then your sensor surface should be hydrophilic, means low contact angle. You have to make. Okay. So how you will make it? So there is some process I will teach you. One process you can make, that is first step. Means slowly. Hydrophilic means the less polarity, right? Your surface less polar. So you have to dip, like you have small sensor chip, right? So you have to dip in a solution that polarity is less, like you can use some acetone, solution. You have to dip all the chip and you have to sonicate it. Sonicate it for 10 minutes. You have to sonicate. Sonicate. Do you know the sonication, guys?
No, sir.
Sonication, it is kind of a sonicator. It is okay. I will let's show you then. Yes, so it is kind of this kind of things. So it is a sonicator. So what you will do after dipping your electrode in acetone, you have to put in this machine, a sonicator. So this is a water bath sonicator. You have to fill the water here and you have to take all the electrode in a beaker that contain the acetone, and you have to put inside this sonicator. Sonicator is the count of sound vibration it will generate. By this, it will, some external force will get on your sensor surface, so that slowly all the contaminant they will detach from the sensor surface. So what kind of contaminant? Some oily contaminant, some other external contaminant, they will be detached from the sensor surface. This is the sonicator. Or I can show you some video how the sonicator works. This is the difference. This is actually, there is so many different. This is called the probe sonicator. Actually here, this is the probe sonicator. The different kind of sonicator available. They're not I'm telling. This is the different sonicator. From here they can generate the sound vibration so that we can remove all the contaminant. So I'm saying here bath sonicator. Bath. Wait. Bath sonicator. This price. No. I think at the end of this video they will show.
Do not ever use alcohol, gasoline, or flammable solutions. Doing so could cause a fire or explosion.
Yeah. So you can see some sound here. You can hear. Can you hear some sound? No,
Yes, sir.
Yes, sir.
See like this. So they're generating some sonication. So like this sound, they will help to remove all the contaminant from the surface. So okay. So then how will remove how will generate your sensor surface hydrophilic, right? Okay. So makes the sensor surface hydrophilic. So first use the acetone and sonicate it for 10 minutes in a beaker. In a beaker, just take all your sensor, dip it fully in acetone, sonicate it 10 minutes. So the organic contaminant, they can be removed. Again, so generally that last video was seen, generally some flammable material you should not keep for the long time. Like don't keep like a 60 minute, not like this, 5 to 10 minutes. Then you have to use the ethanol. Then don't put long time, at least 5 to 10 minutes, just leave the 10 minutes. Then after removing the acetone, then again you have to dip all the electrode in ethanol solution for 10 minutes. Then again wash it, then put water solution. So slowly we are going through acetone, ethanol, water, means we are increasing the polarity of the solution. Then again sonicate it for 10 minutes, water. Like this way, you can remove all the contaminant from the surface and you will generate more hydrophilic group on the sensor surface, and your surface will be more hydrophilic. Okay. Clear? Up to here? Okay.
Yes, sir.
Okay. Now you can see one chemical, APTES. Okay, so this is a I'm not mentioning her full name. No need to remember full name. So if you just search in, I can show you also, but no need to remember. APT is one chemical that contain one amine group. So this amine group we want to functionalize on the sensor surface. So if you draw this amine group contain molecule on the surface, so you will get like this linker on the surface. And your surface will be functionalized with amine. Okay. This molecule that contain the amine group, that will be immobilized just by drop casting method, and you will get the amine group on the surface. Now, basically your final goal, you want to immobilize a primary antibody that contain immobilize this amine. Last time I taught you like carboxyl group and amine group, they can be functionalized. But how amine? For this, you have to add some extra chemical that is called glue, means glutaraldehyde. This full from glutaraldehyde that contain these two functional group. This is aldehyde functional group. So basically, on the surface, then you are getting this COOH. This is called aldehyde functional group. Now, if you add your capture antibody, these aldehyde and amine, they have very strong binding affinity. They can bind, and you will get a primary antibody coated sensor surface. So first thing, you have to clean the sensor surface with acetone, ethanol, and water mixture. Then you have to make the surface hydrophilic. And then you have to add the APTS, make the amine group. Then you have to add the glutaraldehyde, make the aldehyde group. Then add the primary antibody on your surface. You will get primary antibody coated sensor. Your sensor is ready for detection now. Okay, students, any questions here? Do you have any questions, guys? No?
No, sir. No, sir.
Okay, fine. See, just this one, you always have to you keep remember that cleaning the sensing surface is very important before using them. While you receive from any company, any industry, you have to clean them. So by using number 1, number 2, number 3 steps after sonication, if you feel that your sensor surface is not that much hydrophilic, how you can measure not that much hydrophilic? You can measure the contact angle. So contact angle kind of thing, you can drop 1 drop of water. So if your water is like this and contact angle very high, it means your surface hydrophobic, not hydrophilic. But if your sensor surface, I mean, if the drop can spread easily on your sensor surface and you are getting low contact angle, it means your sensor surface water spreading easily, right? It means your surface is more hydrophilic, less contact angle. But if you see that your surface after going through 1, 2, 3, still your surface is not that much hydrophilic, you can add one more step. Number 4. Number 4 step is a mixture of ammonium hydroxide plus hydrogen peroxide plus water. This ratio is 1:1:5. Ammonium hydroxide, hydrogen peroxide, and water. 1:1:5 ratio you can make. And then in a beaker, just put this solution, then put all your sensor chip, and you can sonicate this one for 10 minutes again. Or you can heat this solution around 70 degrees Celsius for around 10 to 20 minutes. You can heat. These 2 things you can do. After these steps, just wash with water all the sensor surface, and you definitely will get very hydrophilic chip. Contact angle will be with theta will be very, very low. Okay.
Sir, why don't we use it in the first place?
Why?
Don't we use this solution 1:1:5 solution in the first place?
No, I'm asking if you need very strong cleaning. Maybe sometimes your surface that you receive from any industry that is not that much contaminated, maybe 1, 2, 3 steps will be enough. But if you check the contact angle, if you see that no, still not clean yet, you need some stronger cleaning, then you can process the 4th step. Otherwise, no need to go to the 4th step. This is a stronger cleaning method. Okay. Clear?
Yes.
This is called kind of soft piranha solution. Anyway, you don't need to remember the name. Just you can remember that ammonium hydroxide, hydrogen peroxide, and water mixture. If you put 1:1:5 ratio and dip all this electrode and clean it for 10 minutes sonication or heating 10 to 20 minutes in 70 degrees Celsius, then almost all the contaminant can be removed. Not only that, your surface will be much hydrophilic and lots of hydroxyl group will be there.
So, sir, if theta is low, then it is more hydrophilic.
Right. See, means it is the water attraction. I mean, water. So if you drop 1 drop of water on a surface, so this is a sensor surface. Yeah? You drop a water. So if it is like this, it is not spreading easily, right? It means it don't have much attraction towards water. That's why it is not spreading. that it is the hydrophobic surface. But if your surface, if you drop 1 drop of water, if it's sprayed easily, it means its water attraction, towards the water, it's very high. That's why you are getting theta is very, this contact angle is very low. But in this case, this contact angle is very high.
Okay, sir. Okay, sir. Thank you.
Okay, so this is one bioimmobilization method. Now come to the final. See, this is the final chip. You can see here. See here. So just don't think drain this kind of, just leave it. This is later you can remember, just don't think this one. So this is your transducer, right? This is your transducer. On this transducer, you add APTS. So APTS means this silicon-based compound. It has one amine group, right? Then on this amine group, you add glutaraldehyde, glutaraldehyde. So it has one CHO group at the function end of this. So on that CHO, if you add primary amine group, that is come from antibody, it will bind. Right, so up to here, it is your sensor surface that is ready to go for detection. So this is your target. Now, if you add your sample, if it contains the target, it will bind, right? This step by step, all the fabrication step. So first, you are adding some linker that basically help to bind the primary antibody. After adding primary antibody, this surface is ready for detection. Now add your target. Clear? All?
Yes, sir, it's clear.
Yes.
Yes, sir. Yes, clear, sir.
Yes, sir. Yes, sir.
Okay, I think I taught so many things today. It will be too much load for you, right? Let's read this one and try to understand all the technology that I'm going to teach. And I already taught you why we want to use this nanotechnology and how we can bind the different kind of biological molecule, and what is the amplification method, how we can amplify. Just try to understand. The next class I will teach you again some other new technique, some binding method and some electrochemical method that I will teach you next class.
Okay, sir.
Okay,
Okay, sir.
Okay, sir. Thank you, sir.
Wait, I can take now the attendance. Just wait. I can take the attendance yours. So how many you are today? Almost 43. How downloaded your attendance? Okay. So is Akhilesh Kumar, is it good? This class, everything fine? Can you understand?
Yes, sir. Yes, sir.
Okay, very good. And Bimalandu? Hello, Bimalandu.
Yes, sir.
Is it okay? Everything?
Yes, sir. It is okay, sir. It is very interesting.
Okay. Devanjali? Devanjali De.
Yes, sir.
Okay. So one thing, yes, I'm always asking you repeatedly. So please, if you have any question, don't hesitate to ask, because you should understand the basic part, right? Now I'm teaching you the very basic.
Yeah, yes, sir.
Very basic things. How you can how you can make the surface from the primary level. You receive some chemical electrode, the bare surface. Now you are going to modify by nanoparticle, by antibody, right? Some very, very basic part. So don't hesitate to ask me. If there is not clear, anything not clear, then final moment when I will going to teach you the final device, that time you may face some problem. Okay, then I can. Diptesh Vishwas. Hello, Diptesh.
Yes, sir.
Is it interesting?
Yes, sir.
Good. Monica?
Yes, sir.
You don't have any question, right? You understand everything?
Yes, sir. I don't have any query.
Very good. Okay, that's all then. We'll again. I will teach you the next topic next class. Thank you. Thank you all.
Okay, sir.
Okay, sir.
Thank you, sir.
Thank you. Have a nice day.
Thank you, sir.
Have a nice day.
Thank you, sir.